Hide this away. All right, ladies and gentlemen, let's talk about cell communication, signaling, regulation. What happens to you're scared of something like this test, which you shouldn't be after you have reviewed this? Okay, so the first thing I want to go through is signaling between cells and the 4 different types that basically pertain to where that signaling is going from. So in all types of signaling, there is going to be a ligand, and there's going to be a receptor, and there's going to be a cell. All scenarios, this is going to be the case. Sometimes there's a channel that releases the ligand, but always a ligand is just a chemical messenger that binds to a receptor and causes a change. So if you are stimulating yourself, some sort of positive feedback, which I'm also going to talk about today, that is autocrine signaling. If it is one cell, like something on the cell membrane of one cell binding to a receptor on another cell, that's direct contact signaling. Now, if the cells are close but not directly in contact, that's paracrine. And then if you were a long distance away, and again, this could be, from the brain to the kidneys, the brain to the toes, brain to the pancreas, depending on where the signaling is going to be, you are going to be endocrine. If it's a gland, some cell in some gland releasing a hormone, that is going to be endocrine signaling, because that's going to be sending it from one location to another, where it's going to bind to a receptor and then cause a change. Okay, I think that's a pretty good summary of that. And this is the overall scheme of any signal transduction pathway. Again, ligand binds to a receptor. Sometimes that receptor is inside the cell, but either way, the ligand is going to bind to the receptor. And that's called the signal perception or signal reception. The receptor will only be on target cells. That's one way you make sure that ligand released from the brain goes and works on the liver instead of works on the stomach cells or the pancreas cells or something like that. Then the signal gets amplified. We're going to talk about that more, but this one signal here can, stimulate several other signals or several other molecules, which will cause a larger and larger cellular response. And it can be one of two things. It can be making a protein, or it can be activating an enzyme, which is like this, where we see in this case, where this is going to be some sort of hormone receptor, because it can go right through the membrane, and hormones are hydrophobic. So it can go right through the membrane and bind to some receptor inside, and then be a transcription factor that's going to cause some gene to be transcribed and then some protein to be made. Alternately, a ligand can bind a receptor, and then that's going to start a phosphorylation cascade, where it activates a relay molecule, or what we often call a secondary molecule, which is going to activate. Notice and remember, kinases phosphorylate other things. So yeah, I'd like to think it's kind enough to activate something. I don't know, that's a horrible analogy. I'm sure I could think hard and find a different one, but kinases activate other proteins by phosphorylating them, because this one phosphorylates number 2, which makes it active, which phosphorylates number 3, which makes it active, so on and so forth, until you get your final cellular response. We're going to talk more about that later. This type, where it's activating proteins being produced by genes, is a slower type. And then this type, where it's activating enzymes, kinase is an enzyme, it's going to be very fast. It's going to be temporary, like blood clotting, or releasing glucose, which we're going to talk about later. Those are cells. Those are goals of cell signaling. Okay, so I talked about this a little bit, but there are 2 types of gene regulation signaling. There's hormones, which go right through and bind to an intracellular receptor. Intracellular meaning inside the cell. and then it's going to go into the nucleus where it can be a transcription factor and cause a gene to be transcribed and then eventually translated. Alternatively, there could be some sort of water soluble. So steroid's going to go through, and it is, hydrophobic, but you could have some sort of water soluble hormone as well, sometimes called peptide hormones. They're going to be released from the cell. They are going to travel through the bloodstream to their target cell, where they're going to bind as a ligand to a receptor and cause signaling there. Now, I'm going to remind you, this little thing, when you see it here, is a cyclic AMP. It's the most famous second messenger, cyclic AMP. And these are just generic proteins that are causing some response where it's going to cause transcription. Remember this, and I'm going to go through the full story of epinephrine again, or adrenaline again, and how it works. Speaking of which, when you were afraid of something, be it a test, hopefully not too much, or a saber-toothed tiger, definitely worth being afraid of, what happens is you are going to, from your kidneys, release epinephrine. So, and again, this is just a sample signaling pathway. There are several types of GPCR pathways, but sometimes it's just like this. You have this fight or flight response that releases epinephrine. It binds to 2, which is the GPCR, or the G protein coupled receptor. It's a receptor. You can see it binding to the site here. Then it's going to release an activated G protein, which is called that because it's got GTP, kind of like ATP. This activated GTP is going to go activate this guy, adenylyl cyclase. And again, you probably don't need to memorize these things, but knowing them isn't going to hurt. The big idea is this adenylyl cyclase is going to take ATP, this guy here, or this guy here, which is a little bigger, and convert it into cyclic AMP or CAMP. Cyclic AMP or CAMP, A here. And this is our relay messenger or second messenger, because it's going to activate all these enzymes, usually kinases, in order to eventually get your response. And this is, again, this is the overall what's happening. But don't forget that when a ligand binds to its receptor and it activates a protein, that protein can activate several other things. In this case, it could be an adenylyl cyclase that activates or that creates lots of cyclic AMP, lots of second messenger. And then those can act on several other proteins by phosphorylating them, several other, and those can act on several others. So you see this, doubling this amplifying of the signal, where one little receptor binding can cause a whole bunch of things to be activated, because once it's activated, it can activate several other things. So I'm going to stop here. I've already talked through a lot, and I'm going to see, are there any questions? Hopefully I'm kicking off some of the...
What's number 4 in this picture you're showing again?
Like this guy here.
Oh, like the very last picture, like the
this guy.
numeral 4.
This guy is a protein called adenylyl cyclase. Actually, I'll open Hovercam and I'll write it out for you. It's not something that you necessarily need to memorize, but it couldn't hurt. It's called adenylyl, I believe. It could be wrong. This could be an A, cyclase. But what it does is converts ATP to CAMP, which activates other proteins in a cascade. And it's called a cascade, kind of like you might have like a cascading waterfall where, water rolls over this rock, which rolls over this rock, which rolls over this rock. And that's what the kind of reminded scientists of this activates this, which activates this, which activates this. But again, 4 is an adenylyl cyclase. If you know it as a protein that activates cyclic AMP, you're okay. Any other questions? Okay. All right. So we're going to now kind of switch gears and talk about cell regulation. Remember, in mitosis, that's just this phase here, and it's PMAT. But you've talked about probably that quite a bit. I find what students don't think about a whole lot is the rest of the cell cycle, and what's happening and how it is checked. So this whole part here is called interphase. Everything that is not M phase is interphase, but it can be broken down into 3 parts. There is G1 phase, which is like growth 1 phase. There is the S phase, which stands for synthesis of DNA. This is when DNA gets copied. And then there's G2 phase, which is the phase in which it's like the second growth phase. They used to think it was a gap phase, and it's when things are growing and fully getting ready for division. So if a cell is going to divide, right, it's going to be checked. And there are 3 major checkpoints. Checkpoint 1 here is going to check size and make sure it has enough room for DNA synthesis. Check nutrient levels, check growth factors, which are just proteins that essentially are going to tell. They're kind of like transcription factors, and they're going to tell the cell what to do. If there is a no signal, essentially they don't have the right size, nutrient levels, or growth factors, they're just going to go into that G0 phase. If you remember, we kind of show it out here where the cell is just hanging out and doing its thing, doing metabolism, being a nerve cell, being a liver cell, what have you. And it's going to hang out here until either it gets large enough that it will enter this, or it'll just stay here permanently, like nerve cells do. Checkpoint 2 is after synthesis of DNA. So it's after DNA replication, and that's going to be mostly what gets checked here at G2. And then M phase is going to be, or the M checkpoint or checkpoint 3, make sure that all the sister chromatids are attached. So I talked about briefly this week about non-disjunction, and where the chromosomes don't get separated into the right parts of the cell. If you have cell division and we have 46 chromosomes, say it's meiosis and it's going to get sorted into 23, or you have the 46 chromosomes that are doubled and you want a full cell with half the right copy. If they're not attached in the center correctly, here, maybe I'll draw you guys a figure as long as I've got my hover cam up ready to go. Say we're at, we're at PMAT, and the cell wants to divide, and we've got these chromosomes here. We're at metaphase, right, where they're all lined up in the middle, and you've got chromosomes. This one has its, spindle fibers that are connected right. And this one has its spindle fibers that are connected right. But this one in the middle, let's say it's only connected on this side and this side never connects all the way. Well, when the cell divides all the way, you would get, half of this one and half of this one. But down here, you ended up with half of this one, 2 copies of this middle chromosome, and then again the other half of this one. So you would end up with too many chromosomes in this cell and too many chromosomes in this cell, which is what the cell doesn't want. So what it does, it has a checkpoint right here to make sure that's not the case. If it does have a problem with spindles attaching and it's ready to move on, it will initiate apoptosis, and it'll make the cell die because something is wrong with the copying mechanism. Because if you don't do that, you end up with cancer, which you don't want. Okay, so that's all fine and dandy. Okay, there's checkpoints, but does that mean that there's little, people there that are, checking and holding up gates and making sure the cell doesn't move on? No, that's not how cells work. What does happen is that you have the CDKs, cyclin-dependent kinases, that are always present throughout the cell. And as time goes on, your cyclin levels, this protein, will build up. And when it builds up to a certain amount of cyclin, it will bind to the CDK, and that's going to tell the cell, okay, good, we can move into the M phase. This is only going to happen if, again, G2 checkpoint, that checks for DNA copying, and so your cyclin levels are only going to build up if you have the right copies of, two copies of your DNA, so you don't get the wrong amount of DNA. This MPF is called mitosis promoting factor. It's just important to know that they discovered this MPF before they knew it was two different parts. That's why it's named this. They go, okay, I know what mitosis is, and oh, you need to have this protein complex in order to go into M phase. But they eventually later discovered that CDK was always present, and cyclin goes up over time until it reaches its maximum and is enough and high enough in order to allow the cell to go into M phase. And then after that, the cyclin breaks down and MPF becomes inactive because the CDK is always present, but the cyclin is not. Okay, so I'm going to stop again to see if there are any more questions. Not hearing any. Okay. I guess this is just an overall schema. Again, I want you guys to think through it. In fact, why don't I have you guys just take a second and try to name at least 3 of these numbers here, and try to name at least 3 of these numbers here. You can write them down on paper. I won't make you tell me them, but this is a good check for understanding, just to see if you remember. Give you maybe 10 more seconds. Okay, so hopefully you noted that number 1 is M phase, number 2 is interphase, number 3 is G1 or growth 1 phase, 4 is S phase, and 5 is G2 phase. 6 is mitosis, whereas 7 is cytokinesis, where the cells actually separate from one another. And then this is just an overall scheme of mitosis. Again, it's PMAT, prophase, metaphase, anaphase, and telophase, during which cytokinesis is occurring at the end of. Okay. This is again just another figure that I liked. There's a DNA damage checkpoint here. There's a this is one that we don't talk about too much. It's not a major one. This is a big one. The DNA replication checkpoint is the one that we were just talking about. Here's another one that we don't talk about too much. And then this is the big one that I talked about, where we want to make sure that there's not non-disjunction occurring, which is it here. And I'll share these slides with you. So feedback, I wanted to talk about negative feedback. Negative feedback is the most common type of feedback. And it's really just the idea that your body has a certain set level or a certain medium level of many different things in your body: your temperature, your glucose level, your amino acid level, just many things where there's an optimal level that your body wants to stay at in order for everything to work well. So let's say in the case of blood glucose, if you have too much blood glucose, then that's going to stimulate the pancreas, that the blood sugar levels are rising. And then beta cells in the pancreas are going to release insulin into the blood. They're going to, the insulin's going to bind to receptors, and it's going to tell the liver to take up glucose, basically convert glucose into glycogen, and tells the cells to take up glucose too, so that instead of having all this blood glucose in the level, it's now stored in the liver and stored in the blood. And then because of that, your blood glucose levels are going to decline back to that preferred level. But let's say you're hypoglycemic, meaning you have your too low of a glucose level. Well, that in turn is going to stimulate the alpha cells in the pancreas to release a protein called glucagon. And glucagon tells the liver to do the opposite. It acts kind of like epinephrine, but in more of in a different way, because remember, the same, it's going to bind to a different receptor, because the ligands are usually pretty specific to the receptor. So the liver breaks down glycogen and releases glucose, and now your blood glucose level is back up again to a good level, and hopefully you become less hangry. I know I get hangry when my blood glucose levels are too low. That's negative feedback. Basically, it's always returning back to a normal level. Whereas positive feedback is where it's a cycle that increases itself. And sometimes, our cell signaling pathways can be thought of like a positive cycle, because, this thing activates this thing, activates this thing. So it could be those. Blood clotting is a, positive feedback, because, thrombin, if you remember, is going to activate, earlier things in the pathway. But just a very common common example that they like to use is if you have, let's see here, you have contractions, right? And at the end of pregnancy, you're ready to give birth to the baby. The contractions cause the release of prostaglandins that are going to release oxytocin, which stimulates the brain to secrete more oxytocin. The oxytocin induces more contractions, then we get more prostaglandins, then we get more oxytocin, then we get more oxytocin, then we get more contractions, and so on and so forth until the baby is born. So positive feedback is typically going to be something where it's going to stimulate something earlier in a pathway or in a cycle, when you want something to go from one state all the way to another state. Whereas negative feedback is going to be where you want to return to like a normal state. But when a baby is ready to be born, the baby wants to go all the way out. You don't want half born. You don't want a middle level of born. You want a complete finishing of the birthing, I guess. So yes, this is why it's a positive feedback. And I think that's the last slide, and I will ask questions again. Does anybody have any questions? Remote stirs?
Do you know anything specific in like the two examples you just gave for positive and negative?
This one comes up, is illustrated, but you're not going to necessarily, if they ask about beta cells or they ask about alpha cells, they will tell you what is being relayed, but it's not like they're going to say, what does insulin do? You know what I mean? They're going to say something like, insulin is found to lower the glucose levels, and whereas glucagon, another protein from pancreas cells, is going to raise it. This is demonstrates what type of mechanism, and you would say something about negative feedback. Whereas if it was a pathway like giving birth or clotting, where one thing stimulates something else earlier to cause a greater effect, you see, the stimulus causes more stimulus essentially, then it would be a positive feedback. But that's really all they need to know. Don't need to worry about memorizing prostaglandins or oxytocin or things like that. Okay. Any other questions? In future biology classes, you might need to know these things, but in this one, you don't. My wife talks about these both a lot in her physiology class. Okay. So if there are no other questions, I'm going to stop recording.